Okay, fantastic. I believe we have over 500 people registered for this seminar series from across the world. So good morning, good afternoon, good evening to everybody who's joined us today. This seminar series is the first Human Cell Atlas seminar series for the consortium at all to have this online format. So we're super excited about this and very grateful to both the speakers and the attendees and the panelists. And the history behind this is that, of course, we were going to have a Human Cell Atlas developmental biology meeting in Toronto, kind of hosted by Gary and the committee, that couldn't take place in person. And so in the meantime, until we can all get together in person, we're running this seminar series. And today's topic is the lung, and I will now hand over to Aviv for a couple of extra points before we get started.
So I'll give you a little bit of a sense of the agenda for this one and future ones. So the idea is that each session is going to have talks from two perspectives on a topic, like basic, say, research and clinical applications, or wet lab approaches and computational approaches. And this would allow us to link researchers who are already working in HCA or in related single cell mapping projects with others who might not yet be connected, but where everyone shares this passion and interest in a developmental atlas. And developmental atlases pose some unique challenges, as well as some challenges that are shared with the broader context of the HCA. And then we will have a panel discussion at the end to bring in additional perspectives. We really hope this will help energize people and help with networking people together, just like we have in traditional face-to-face meetings, but virtual. And it does have the silver lining that in a face-to-face meeting, we're usually more limited in the number of participants. And in this virtual one, as Sarah said, almost 500 people registered. And in fact, people are still trickling in. So not so trickling, I would say flowing in. And so I also want to point out the speakers today. They're Gloria Pryhuber and Mark Krasnow. And Gloria is also a member of HubMap, and Mark is an active member of HCA. And our panelists are Helen Firth, Muzlifah Haniffa, Dana Pe'el, Gary Bader, Sarah, and myself, under the great guidance of Bruce Aronow. And the next seminar is going to be on spatial transcriptomics, and will be with Matt Nielsen and Ali Erturk in a TDB date, so look out for it on our website as well as on Twitter. And finally, if you want to ask questions, you need to use the Q&A function at the bottom of your Zoom platform. I'm sure many of you by now are old hands at that, and we'll do our best to answer any and all questions as much as time permits.
Oh, you can hear me now? Okay, great. Just a couple words about Gloria. I've had the privilege to work with Gloria on LungMap 1 and now LungMap 2 for a while now, and she's just an amazing person, and you'll get that in listening to her. She's trained as a pediatrician and neonatologist, and part of the human development, the challenge in looking at processes in humans, healthy and sick, is in dealing with the people who could provide samples. And so Gloria's had this tremendous experience, and she working with parents of kids who are in the neonatal intensive care unit, of dealing with their diseases, and the issue of how do we get them to work with us and try and help other kids who may be suffering from similar diseases. And so, her empathy and understanding of that whole process is something that I think really can give us a lot of insight into how we can work with people in different groups to get better samples into the area of human development for single cell atlas. So with no further ado, thank you so much, Gloria, for offering to give the talk today.
Well, thank you, Bruce, for inviting me, and everyone at Human Cell Atlas that was responsible for asking me to give this kickoff talk for the developmental series. I'm very excited and honored to do it. I also want to acknowledge the funding organizations. As was mentioned, I'm a member of LungMap, phase 1 and phase 2, and of HubMap, recognizing the National Heart, Lung, and Blood Institute for their support, and also my parent institution, University of Rochester Medical Center. So thank you, and let's get going. So I'm trying to get that out of my way. Okay. So already introduced, I understood this to be a series of mini conferences mapping human development from conception through childhood at the single cell resolution, the what, the why, and I will give you more of a clinical background as a neonatologist. I know that I'm talking to a diverse group of folks, all with different backgrounds and different interests, and I hope to say something at least that will touch on each one of those interests. I'll have to go relatively quickly to stay within my time, however. So and in my role as a neonatologist and someone who is faced in taking care of children and their families when they're faced with complex lung disease, I'm going to try to give you some perspective as a physician scientist on normal and abnormal fetal lung development and postnatal lung maturation. I'll touch just a little bit on genetic mutations known to cause perinatal lung disease. Many more, we're relying on you to help us to determine those polygenic causes as well. I'll talk a little bit about the response of the developing lung to extrinsic perturbations and iatrogenic. I do things to these lungs that cause problems in keeping children alive. I'll give you a little bit of a sense of the timescale about the development and how these diseases and recovery or lack of recovery develops in people. And then we'll touch on how to go about collecting and profiling for human lung development growth studies based on our most recent experience from the University of Rochester. And then I'll try to give you a little bit of my perspective as a physician scientist on how single cell lung developmental maps will eventually help us in the clinic. So perspective on development maturation. For us to be able to be sure that we have everyone on the same wavelength here, I felt it was necessary to go through a little bit about the complexity of the human lung and how it develops. And so we all think about the lung, its primary function as gas exchange, also plays a very significant role in immunology, in our protection and interaction with our environment, with an extensive mucosal surface that interacts with the environment. The lung is usually thought about as being arranged around the central airways. And so the airways are a series of dichotomous branching tubular structures, the first section of which, the first 15 or so branches, are really conduits for air to get it in and to get it back out. And then the more distal branching structures and then alveolar structures are really the gas exchanging units. So in order to be able to exchange carbon dioxide and oxygen, we also need a matching vasculature for the airway structure. So we actually have at least, we have two vascular systems within the lung. The first being the pulmonary vasculature, responsible for bringing deoxygenated blood in through the pulmonary artery from the right ventricle, bringing it to the capillaries to exchange gas, and then bringing the oxygenated, cleaned blood back out through the pulmonary veins to the left atrium. But there is also, people forget sometimes that there's also a bronchial vasculature, which I think of as being responsible for nutrition and oxygenation of the lung tissue itself. That comes from the systemic circulation from the aorta. Again, it brings nutrition to the majority of the lung tissue. There's also extensive muscularization of all of these tubular structures, both the vasculature and the airway, and that maintains the match between ventilation, the ability to bring air in, and perfusion or VQ match ventilation and perfusion that is absolutely required for normal lung function, and is really where a lot of disease gets us to VQ mismatch. There are extensive lymphatic system within the lung that recovers fluid. The lung is somewhat at risk for developing edema and interstitial fluid. The lymphatics also return immune cells back into circulation. The musculature is heavily innervated, again, to be able to regulate the size of the tubular airway and vascular structures. And then there must be a scaffold that allows for the dynamic movement of the lung. Our respiratory cycle of inflation and exhalation, preventing that scaffold must prevent collapse of the airway. Oh no, phone call right in the middle. Okay, somebody else will get that call, hopefully, from IIAM. I'll talk about them in a minute. Okay, so our radiologists think about the most distal lung as a series of secondary lobules. So a lobular bronchiole and its accompanying artery supply a secondary lobule that these are divided by interlobular septa. In those septa run the pulmonary veins, and composing those secondary lobules are pulmonary acini. In the adult anyway, somewhere between 0.5 and 1 centimeter in diameter. The other measurements here give you some idea of perspective and relative size of these structures. So our radiologists actually do think, they may not call them secondary lobules in their mind, but they do think about those secondary lobules to be able to interpret the radiologic images and determine disease. So in these many unique structures in the lung, from the trachea through to the terminal bronchioles, the bronchi with their cartilaginous plates and their submucosal glands, the terminal bronchioles, respiratory bronchioles, and then the alveoli. These are obviously all composed by many different lung cell types. Classically, we've always talked about 40 to 45 lung cell types. I think Dr. Krasnow will give us, has informed us that we're talking more about 50 to 60, and maybe depending on how you determine a cell type, even more than that. So one of our challenges is the lung is not a uniform structure from proximal to distal. It varies in its cellular structure. So to do single cell biology, we need to be able to optimize cell recovery and performance in single cell assays while retaining some concept of the morphology, the relationship of those cells to physiology, the ability to translate back into how those cells function in a whole organ. This is a developmental talk. And so I wanted to go through the fetal and postnatal development of the lung just a bit. As many of you probably know, the lung begins with fertilization, moving through an embryonic period, pseudoglandular, cannulicular, saccular, and then our more classic alveolarization and maturation up to young adulthood. Just to point out as a neonatologist that our limit of viability now is somewhere around 22 to 23 weeks of gestation. And that puts us very much into the cannulicular and saccular stage. So these are very immature lungs that we're dealing with, and successfully keeping children alive, but not without sequelae. So again, for our single cell biology challenge, we have to consider the lung as a developing organ, even postnatally, that is requiring growth, maturation, and continued repair to keep a healthy lung. Looking at this just a little bit differently, I do want to point out that when we talk about gestation from a maybe developmentalist point of view, we're talking up through 38 weeks of gestation. But when I talk about it clinically, I add on the 2 weeks from the last menstrual period up to where fertilization can occur. So full term for a clinician is about 40 weeks gestation. And so this diagram, again, shows you the stages of lung development, our point of viability up through gestational weeks. Surfactant is a material that is produced on the surface of our airway, especially the distal alveolar structures. This allows or functions to decrease surface tension, so that this lung, when it partially collapses or completely collapses, can be opened easily in the next inspiration. Without surfactant, the structures collapse and become very non-compliant, impossible to ventilate unless breathe spontaneously. This diagram also allows me to remind us that the airway forms from more proximal to more distal, starting with a tracheal development in the embryonic period, moving through the branching of the bronchi, bronchioles, terminal bronchioles, up to about 18 weeks of gestation, and then further development of the gas exchanging structures, respiratory bronchioles, alveolar ducts, alveolar sacs. Again, revisiting this development in a little bit different way, now looking at the structures that, or the events that have to occur for normal human lung development, starting again with the trachea and the pulmonary veins and arteries, moving through closure of the diaphragm, airway branching, the growth in length and width of those airways, and then development of the saccules, alveoli, and gas exchange. The diagram at the top reminds us that the cellular structure does change. This focuses on the epithelium that goes from a more cuboidal structure to one that has cuboidal type 2 cells producing surfactant, as well as very attenuated egg-like type 1 cells that form the majority of the gas exchanging structure of the alveoli, and interact very closely with the endothelium of the capillary with its red blood cell capable of exchanging gas and carrying oxygen. Okay, so each one of these stages has a potential for error that as a neonatologist I end up having to work with when babies are born. So very early on, the development of the trachea separate from the esophagus can go awry and we develop fistulas. The formation of the tracheal rings can be complete instead of C-shaped rings. This causes abnormalities in the function of those airways. Alveolar capillary dysplasia with abnormal misplacement, particularly when there's misplacement of the vascular structures. In the pseudoglandular, when the diaphragm should be closing, if it doesn't, we end up with a congenital diaphragmatic hernia. If the amniotic fluid is not maintained well in this period of airway formation, we end up with a severe form of pulmonary airway hypoplasia. This might be due to rupture of membranes or to kidney, urinary tract failure. Further into the development of the respiratory status with severe in utero growth restriction when the placenta is not working well and growth overall is compromised, or where there are congenital airway malformations occurring. We see obviously those sequelae when the babies are born. We also have congenital malformation or mutations of surfactant proteins, as well as babies who are born in this premature stage develop a chronic lung disease of bronchopulmonary dysplasia. We also have abnormal immature control of breathing in those preterm infants. So the neuroinnervation of the lung is something that we really don't understand well enough yet. In the preterm, we have abnormal airway and vessel reactivity that causes significant neonatal compromise. And so, of course, as we're going into childhood and adulthood, we have complications of lower respiratory tract infections, asthma, and pulmonary hypertension as examples. So these two images of beautiful immunofluorescent chemistry, both from Cincinnati and from Marshop in Rochester, just demonstrates again the cellularity of the alveolus with capillaries, endothelial cells, very tightly coordinated with the respiratory type 1 alveolar cells, the type 2 cells producing surfactant. And then to the right, just demonstrating some of the cellularity and the progression of cellularity as we come down the airway of the bronchiole to give way to the alveolar cells. Okay, so just a little bit about response to extrinsic perturbations and iatrophic interventions. And what I'm going to just mention here is that disease of bronchopulmonary dysplasia. So this is the histology of a normal term lung into the alveolar stage now, where we have thin septa, increasing surface area for gas exchange. Excuse me. When a baby is born in that late canalicular or early saccular stage, in less than 29 weeks gestation in particular, although we can maintain them with ventilators and oxygen, their lungs do not continue to mature properly. So we talk actually about a developmental arrest or alveolar arrest of alveologenesis. In addition, these lungs become injured by oxygen and ventilation. So we have fibrosis, we have capillaries that are not very well coordinated with the airway, gas exchanging epithelium. We have hyperplasia of type 2 cells, shown here by surfactant protein C expression. And this is at 40 weeks post-menstrual age or corrected gestational age. So this lung should look like this one, but it has failed to develop properly. With our ventilators being better and oxygen being used in less toxic quantities, we're not seeing quite as much of the fibrosis. We're not seeing quite as severe a simplification, but still we don't get the normal alveolarization. The BPD lung remains quite simplified. As you can imagine, the capillary bed is not formed properly, either when you don't form normal alveolar structures. And this just shows you by CT scan and by MRI that we have both macro and microcystic abnormalities in BPD. This is in a extremely low gestational age birth, now at 40 to 50 weeks CGA. We do have a differential diagnosis for the sequelae of of premature delivery of BPD. So we now know about a series of childhood interstitial lung diseases, some of which we know the genetics of, for example, acinar dysplasia with TBX4, FOXF1 with alveolar capillary dysplasia with abnormal pulmonary veins, the surfactant protein gene mutations, mutations in ABCA3 that prevents surfactant from being trafficked properly, and KX2.1, transcription factor that controls these genes. But we have a number of other congenital interstitial or diffuse lung diseases for which we don't have either genetic or polygenetic knowledge yet, although we know that there is some heritable component to these diseases. So that's up to you, researchers, to help us figure out. Yes, there is an effect on quality of life as preterm babies and normally born people age. So this is just an idea of quality of life in the early adolescence. And children with BPD, actually their respiratory disease does get better, at least temporarily, so that their quality of life may not be much different from control. This is also just to point out the other major disease that's affecting our population, which is asthma, that we really don't treat yet very well, don't understand very well. And a sense of the time scale. So I talked about the perinatal period of bronchopulmonary dysplasia and how severe it can be. But even in those I just showed you by early adulthood, they may be asymptomatic. Children with BPD, their lungs are not normal, even though they become less symptomatic. So this is just an example in a 24-week infant, and then a 25-week infant, now at 3 years of age, that still have simplification of their alveolar structure as compared to lungs at 3 years of age that were born full term. So how do we get this alveolarization to resume properly in the prematurely born infant? That's my challenge to you. This is just at 10 years of age. Again, simplification and evidence of gas trapping and cystic changes in these lungs. And it's not just those that are born prematurely. So we all, as healthy as we might be, we will all have an improvement and a maturation in our lung function. This just shows how we exchange, how we move air in and out. We'll hit our peak though at about 20 years of age, and then we're all downhill from there. Some of us never reach quite that peak of normal aging. Again, those born premature are less likely to reach that peak, and some of us will decline much more rapidly. So how do we understand that aging process? How do we prevent it? How do we reverse it? How do we treat it? That underlies my interest in these mapping projects that we're part of. Okay, so how do we go about collecting and profiling human lung development? And again, I'm basing this on what we've done in Rochester. As Bruce pointed out, we've been part of the Lung Map program now for about 6 years. And as the Human Tissue Core in Rochester, we've developed a Brindle repository. We have now over 270 cases. Most of them come to us through the transplant network as donations, a range of ages, a range of normal growth to abnormal diseased lungs. We do provide these cells and tissues with as detailed as possible clinical information. They've been reviewed by pediatric pathologists, and we will distribute those back out to research centers and to the broader research community. If there's interest, please get in touch with me. And then we have demonstrated that these cells and tissues are of fairly high quality, and they can be studied with the newer technologies that are now being developed. So just quickly, we bring in these lungs through the transplant network, through the organ procurement organizations. They come to us as en bloc lungs, sorry. We inflate them with oxygen, we do CT scans, we can do measurements of airways. Each component of these en bloc lungs are processed, frozen, fixed, embedded, or cellular dissociated. We've distributed and used these dissociated cells. We've sorted them by flow cytometry and done bulk RNA sequencing, also bulk proteomic and lipidomic at PN and L. We and our collaborators have also done single-cell transcriptome work. We prepare them for imaging studies, and our collaborators have also done single nuclear chromatin accessibility and TAC-seq type assays. So this was one of the earliest studies that we did as part of LungMap. This is RNA sequencing on those bulk sorted cells, analyzed at Cincinnati. And just to point out, so sorted into the four major lung types. Each column is a donor lung, ranging from neonatal, newborn, up through adult. And just very briefly, we can demonstrate the progression of gene expression with age in these various cell types. Much to be more fully understood there. We also have been doing progressively more single-cell RNA sequencing. This demonstrates the comparison of 2 one-day-old newborn lungs, in which we've been able to identify at least 17 different clusters. Each of the major cell clusters are identified here. Working with Cincinnati and drop-seq from mouse lung cells, we've been able to, Sumo Bhattacharya has been working particularly hard in integrating the mouse and the human. We found similarities in development, but the majority of the human newborn lung cells are more like a 1-week-old mouse cell. That's consistent with the saccular versus alveolar histology that we see at birth of mouse versus human. This is just, again, exemplary work from Cincinnati on drop-seq from both a preterm 31-week lung and a 39-week lung, showing progression in the type 2 cells of gene expression from preterm lung to more mature lung with particular genes outlined. So this just gives me an opportunity to point out to each of us as potential donors, but also to help you to understand how important organ donation is to research. The ability to study these human tissues and cells is essential and is unequivocal in its potential to save lives by improving diagnosis and treatment. Without this human tissue research, we're stuck primarily learning from animal models. And yes, I'm a biased clinician, but mice are not human, and children are not small adults. As is probably obvious to most of you, this human tissue research gives us potential for personalized medicine, for gene discovery, for network and mechanism discovery, for drug discovery. All of our potentials for developing organs on a chip, or giving back stem cells, or understanding pluripotent stem cells. These all depend on being able to isolate cells and determine with certainty their identity and their potential functions. We need to know single cells intimately and be able to mimic normal and intrinsic cells. And while I'm at it, what is normal? Until we get down to a single cell level, it's very difficult to know what is normal variation, what is actually abnormal and causative of disease. And then more and more, we identify that adult disease starts in infancy and fetal life. We need to understand these components. As we identify genes and proteins necessary for lung growth and function in the normal patient, we find diseases that are caused by those abnormalities. We can then describe them, define the diagnoses, determine the prognoses, and find ways to reduce disease. And in donation, organ donation, we save not only one child or one person, but many with these gifts for research. So that's just my plea to all of us as a community. So how do I think single, yep, this is my last slide, yep. Yep, thank you. single cell lung development, and where is the value. So growth, development, repair of the gas exchanging machine and the mucosa facing the world is complex in its cellular functions and interactions. If we interrupt that dance, as in a premature birth or a viral infection or asthma-like disease, that can forever change the future of that organ. How do we restart that music without having a note-by-note musical score, that developmental map? I'll just posit that that map or atlas should provide identification of cell subtypes and activity states, the position of those cells, the potential for their structure, their cell-to-cell, cell-to-matrix interactions, the receptor-ligand relationships, and prediction of response to our environment. We're only starting
Gloria,
to understand
thank
metabolism.
You.
Yep.
Gloria, this is great. This is exactly what we wanted you to cover into this, all the different disease scenarios. I think we're a little bit over time. And
okay.
So let's
done.
get back. I think there's some important questions on, excuse me, how we're gonna use single cell genomics to better understand these diseases mechanistically, as well as using them as a clinical tool. It's all of what you're touching on now. Let's get back to that from the panel's perspective after we hear from Mark.
Perfect.
Thank you so much.
Thank you. Yep.
And so, Mark, also thank you so much for agreeing to give this talk here, just as your new paper is hot off the presses, and the data looks absolutely spectacular. You guys have done such a great job on this, and it's a recapitulation of so many beautiful things that you've done in your career. Mark is a professor of biochemistry, served as the chair of biochemistry at Stanford. And I think that the way that single cell genomics has inspired us all to be here in this meeting, it's just such a privilege to see this data, to understand biological systems in this way. Mark's had that in his vision coming up into this whole area. So it's really great that we get to hear from him at this stage, at this junction going forward. So thank you, Mark.
Thank you, Bruce. And thank you for the invitation to present in this new and innovative COVID-mandated format. I am going to, let's see here, talk about our efforts towards a comprehensive molecular cell atlas of the human lung. And by that, I mean all of the molecular gene expression, that is cell types, their full expression profiles, their correspondence with the classical histological cell types, and their location, structures, and neighboring cells in the lung, as Gloria just told us. And what I'm going to focus on today is three aspects of constructing such a map, and then three uses. And that includes the discovery of new lung cell types and new functions for lung cell types. Then talk about what cell types are directly targeted in disease and how are they altered, in particular focusing on COVID for most of the talk. And then how does each cell type arise during development and change during aging, as is the focus of this seminar series. Now, we had been studying lung development, stem cells, and diseases like lung cancer in mouse models for a number of years, and constructing mouse lung cell atlases and mouse lemur full organ-wide cell atlases. But our work on humans began about 2.5 years ago with a very personal note. And my colleague and biochemistry professor and good friend, Jim Spudich, came to me one afternoon about 2.5 years ago, and he told me that he had lung cancer, and he was going in the next morning for surgery, a therapeutic lobectomy. He asked me if I could use the lung and the lung tumor, and because he knew what we had been working on in mice. And after I recovered from the shock that my good friend had cancer, I said, I think we can. And because we had been working with mouse and mouse lemur tissues and lung tissues for many years and for a couple of years at that point, from a cell atlas perspective, we were able to rapidly mobilize what I call the Human Lung Cell Atlas Rapid Response Team. And that's several people in my lab. Ahmad, Kyle, and Astrid have been leading that. Rahul Sinha and Irv Weissman's lab. And importantly, Steve Quake and his remarkable team, Norman F. and their colleagues in the Chan Zuckerberg BioHub, as well as a number of other faculty and colleagues here at Stanford, including clinical colleagues and Jim Spudich himself. Well, the next morning, 12 hours later, after Jim appeared in my office, Ahmad, Kyle, and Astrid were at the surgical suite, and they recovered Jim's resected lung, including the tumor sample, but importantly, as you can see here, 2 different samples, peripheral samples from his lung. And subsequently, over the next year or so, we obtained therapeutic lobectomies and surgical lung resections from 2 other patients, which were sampled in these positions of the lung, as you can see here. And for each of these samples that we collected, we brought the cells directly from the surgical suite to the laboratory and immediately began processing the fresh tissue. We did enzymatic digestion and mechanical dispersion of each of these tissue samples. And then we separated the cells by either magnetic associated cell sorting or fluorescence associated cell sorting. And we separated them into the major tissue compartments, endothelial, immune, epithelial, and stromal compartments, so that we could balance representation of the analyzed cells among all of the major cell compartments, because we were interested in getting a comprehensive atlas. And then we did, on these cells here, we did 10x droplet broad profiling. We did about 20,000 cells from each of the individuals from the sample sites I've noted. And then we also, for a couple thousand cells from each individual, we did SMART-Seq2 plate-based sorting. So to get deep gene expression profiling, to identify even the genes that are expressed at very low levels, which is particularly important for identifying transcription factors, receptors, and other genes that are expressed at low levels. Now, in parallel to these lung samples that I've just described, we did something very similar to the blood samples from each of the 3 patients. So we had patient-matched blood, and we did 10x droplet-based sequencing and SMART-Seq2 plate-sorted sequencing on those samples as well. And in addition, as I mentioned, each of these patients had a focal lung tumor. It was a therapeutic lobectomy. And we did similar analyses of the tumor-associated samples, but I'm not going to talk about those today. I'll talk about those at another Cell Atlas meeting. What we did then was, after the single cell profiling, is we then identified the cells that were from each of the major tissue compartments by their expression of these canonical tissue compartment markers indicated here in the 4 colors. And so what we were able to do is to computationally separate out all of the cell types from each of the 4 tissue compartments, and then to cluster and subcluster those cells iteratively, as shown by the scheme here. We took one of the cell types, for example, the epithelial compartment, rather, from one of the individuals, and then we computationally clustered those cells, and then did a differential expression test to identify the genes that are differentially expressed among the clusters, and asked whether those were biologically meaningful genes. And if they were, then we took each of those individual clusters and clustered them again, subclustered them, and continued this process until the differentially expressed genes were not biologically meaningful. For example, if clusters were being separated by their expression of stress-induced genes or mitochondrial genes or other genes that we thought were unlikely to be biologically meaningful. And then, after we had clustered each of these tissue compartments for each of the 3 individuals, as I've just described, we then merged each of the cell types or the cell clusters that we had identified in each of those tissue compartments without any batch correction algorithms, so that we could maintain the true diversity of those gene expression profiles. And after doing so, with the merged from all the data from all the different tissue sites from all of the 3 different patients, we identified 58 different lung molecular cell populations, or molecular cell types, as I'll call them. Now, we were very interested in relating these 58 molecular types to the canonical cell types that Gloria mentioned in her talk, of which there were 40 to 45 that were known, as she noted. We compiled an exhaustive list from the literature of the 45 known human lung cell types listed here by their tissue compartments, their estimated abundance in the lung, and you can see how there's a huge range in abundance from the most abundant cell types, which are 13 to 20% of all cells, down to cell types that represent down to 0.00003% of the cell types. So a huge range of over 10 to the 5th orders of magnitude in abundance. And then importantly, we collated from the literature canonical cell markers for each of those 45 canonical cell types. And for then looking at each of these 58 clusters for the expression of each of the canonical markers, we were able to assign 41 identified clusters representing 41 of the 45 known cell types. That is, all but 4 of the most rare of the lung cell types. In addition, we identified 4 clusters shown here that were similar to a canonical cell type, but differed in the expression of proliferation genes, shown here, indicated by P, or differentiating genes, indicating that these were cell states, that is, differentiating or proliferating states of a known cell type that was next to it. But in addition to these known cell types and these cell states in the lung, rather remarkably, we identified 4 new molecular cell types in the lung. And because we had now this rather comprehensive set of molecular types in the lung, we were able then to use the atlas to computationally identify the best, that is the most selective gene and protein markers for each of the cell types, as indicated here, which will be useful for monitoring each of these lung cell types, for example, in pathological samples, and also when we restricted just to membrane proteins, to be able to isolate each of these lung cell types, for example, by fluorescence activated cell sorting. And so we've used these markers, like the ones shown here, to localize each of the lung cell types within the lung, and particularly focusing on the new cell types, and also to use membrane markers to begin isolating and characterizing each of these cell types, including, and of course, especially the new lung cell types. So with these analyses, what we were able to do is localize in the lung all of the new cell types and establish their relationship, their cellular relationship and spatial relationship to all of the existing cell types to create, as shown below, this new molecular cell atlas of the human lung, version 1.0. Now, what I'm going to do for the rest of the talk is I'm going to focus on the alveoli and the new cell types in the alveoli and their relationship, their functions, and their relationship to the disease. And I'm going to do this because of COVID-19, and that is that the alveoli are thought to be the critical target in COVID-19. That is the target that initiates the respiratory demise and not uncommon death of these serious COVID-19 patients. And the idea is that the virus, like air, makes its way through the conducting airways, the bronchi, until it reaches the alveoli, infecting and proliferating perhaps in a number of cell types along the way. And then when it reaches the alveoli, it starts to target the particular cell types in the alveoli. In particular, here shown an alveolar type 2 epithelial cell, which it's believed to infect and gain entry and to initiate the destruction and respiratory demise. This is the site where the clinical symptoms, the pathology, and the CT findings are focused in those individuals with severe COVID-19. Now, the first, I think, or one of the first great uses of the lung cell atlas was exactly in this context, that is, was identifying the cell types, like the alveolar type 2 cells here, that are thought to be directly infected by the virus. And that was a nice analysis, and I think a great achievement of this human cell atlas team and a number of other teams across the country and around the world, using an atlas to figure out how is COVID-19, the SARS-CoV-2 virus, getting in and initiating its destruction. Let me just show you how we contributed to this effort. And that is what we did with our cell atlas is to collate a list of all of the known viruses, shown here on the right, and the human proteins that they use as receptors to enter cells. And then what I'm showing in the plot here is the expression levels of each of these proteins, which act as virus receptors, among each of the known cell types now in our human lung cell atlas. Now, the cells that are highlighted here, or the positions that are highlighted in pink, shown here, are of great interest because these are the cell types that the virus has direct access to once it enters the airways of the lung. So these are all the airway epithelial cells that are lining the airways. And then in pink here are the immune cells, the alveolar macrophages that are in the lumen of the alveoli and the airways that the virus also has access to. Now, if you look here, this is COVID-19 virus shown here, and this is its cellular receptor. And you look across here and you don't see any substantial expression, but if you actually look closely, there's low detectable but significant expression, which is easier to see in the plots below. And if you look here at these cells right here, these are the alveolar type 2 cells, which have the most substantial expression, low but detectable, and the most substantial among all of the cells, and represent the most significant fraction of lung cells that are expressing the receptor. You can see that there are a number of other cell types along the entrance pathway, other alveolar epithelial cell types that also express. I also want to call your attention here to these capillary cells, because I'm going to get back to them, these capillary A cells, or aerocytes, which I'm going to talk about in just a few minutes. Well, let's learn a little bit more about these alveolar type 2 cells, their development and their function before we get back to COVID-19. So this is kind of what Gloria described in her talk, is that the lung is built by first building the conducting airways by branching morphogenesis. And then the gas exchange units, the alveoli, begin to form. And then the human lung, that's several hundred million positions in the lung where alveoli are formed. And at each position where an alveolus begins to form, you see the cellular events shown here. So here's the tall columnar airway epithelial cells, 3 cells shown here. And at each position where an alveolus is about to form, the cells start to flatten to form these cuboidal cells. And then some of the cells, like this cell here, will continue to flatten to form an alveolar epithelial type 1 cell, which is the thinnest, flattest cell in the body. And of course, the reason that it's so thin and flat is that it's the gas exchange surface. And so oxygen, which has come into the lumen of the alveolus, has to diffuse across the cell, the alveolar type 1 cell, and then across the endothelial cell of the alveolar capillary cell that is in direct contact, so that the oxygen can reach the blood in the blood cell and then be transported from the capillary back to the heart and to the rest of the body, as Gloria described. Now, this association right here is a particularly interesting one, because not only is the alveolar type 1 cell thin and flat, but so is the endothelial cell lining this capillary, shown here. These cells are directly opposed to one another, and in fact, they share and merge their basement membrane. So they're the thinnest, flattest, tightest connected cells in the body to allow the most efficient passage of oxygen from the lumen of the alveolus into the bloodstream. But this very thin structure also makes it a site of fragility and a number of diseases, including bronchopulmonary dysplasia, as we've heard, in babies and neonates, but also COPD and pulmonary dysplasia in adults. Now, the only other cell type in the alveolar epithelium is these adjacent alveolar type 2 cells here. And these are the professional secretory cells that secrete the surfactant that lines the surface of these thin-walled alveolar sacs and help prevent them from collapsing. And it turns out that these are among the last cells in the body to develop. And because of that, in premature babies, the OBGYN and the neonatologists are trying to keep the baby in the womb long enough so that these cells can develop and differentiate to make the surfactant to keep the alveolar sacs open and keep them from collapsing at birth when breathing begins. Now, there's also, though, as we've learned in the last few years, another completely unrelated function of these alveolar type 2 cells that make them among the most deadly cells in the adult, as I'll describe now. It turns out that these alveolar type 2 cells and the type 1 cells arise during embryonic development from a bipotent progenitor, which either gives rise to the thin, flat alveolar type 1 cells or the secretory alveolar type 2 cells. But then what happens at birth is that the progenitors are gone, at least in mice. And what happens then is that the alveolar type 2 cells begin to function as stem cells that can either give rise to new alveolar type 1 cells, can self-renew, and they remain active and slowly active throughout the life of the mouse. So they have all the classic features of a stem cell, except for one. They appear to be fully differentiated secretory cells. And so they're being referred to as facultative stem cells, that is, stem cells that have a day job, in this case, the secretion of surfactant proteins. And the way this works, as we now understand it, is a subpopulation of alveolar type 2 cells that we refer to as alveolar type 2 stem cells are selected by a neighboring fibroblast that selects out individual alveolar type 2 cells to act as stem cells. But those stem cells remain quiescent until a neighboring alveolar type 1 cell is injured or another alveolar type 2 cell nearby is injured, as Bridget Hogan's lab showed. And those then result in a dying cell that provides an EGF signal that activates the EGF signaling pathway, KRAS and the EGF signaling pathway in the stem cells, and activates then their stem cell activity. Now, normally this is beautifully coordinated, so you have just then enough stem cell activity to replenish the dying cells, like the dying type 1 cell shown here. However, if one of these stem cells suffers an activating mutation in EGF receptor or KRAS or any component of the signaling transduction pathway, the alveolar type 2 stem cells get a constitutive permanent EGF signaling signal when there's no EGF signaling cell around. And that genetically activated EGF receptor pathway activates and continually activates that stem cell activity. And that initiates lung adenomas that progress to lung adenocarcinoma. So it's thought now that alveolar type 2 cells, and in particular alveolar type 2 stem cells, are the likely cause of initiation of one of the most deadliest cancers, the leading cause of cancer deaths, lung adenocarcinoma. Now, this is all true in mice, and let's get back to the human cell atlas. And as you'll notice here, there is two cell types here. There's an alveolar type 2 cell and a second, an alveolar type 2S, which is one of the newly identified human subtypes. The alveolar type 2 are the canonical type 2 cells, surfactant secreting and expressing the receptor for COVID-19. They're shown here in black. And these AT2S cells, or AT2 signaling cells, resemble in the expression of Wnt signaling pathway components, the AT2 stem cells as mice. And so we think they are likely the alveolar stem cells, as are the mice AT2 stem. And also, these AT2S cells are the cells that most resemble the cancer cells of lung adenocarcinoma patients, like the adenocarcinoma cells of my colleague, Jim Spudich. So these two cell types are here in the lung, the canonical ones intermingled with the alveolar type 2S cells, the potential signaling cells. And let's just go back now and take a look at the COVID infection. So what we imagine is that when the virus gets here and infects the alveolar type 2 cells through the ACE2 receptor, it starts proliferating and destroying those cells. and leading to alveolar cell destruction and death. But not shown here are the alveolar type 2 stem cells, which are starting then to replenish themselves, to proliferate and replenish these. And it's a battle then between the virus infection and the ability of these AT2S or AT2 alveolar type 2 stem cells to replenish that. Now, if the virus gets the upper hand, then the virus starts to destroy the epithelium and breach the epithelium and begin to penetrate and infect the underlying stromal cells, including the endothelial cells indicated here of the alveolar capillaries. Now, I'll get back to that in a minute, but I want to, COVID-19 in a minute, but I want to just talk about capillary cell types, which is now the other side of the alveolar coin. And it turned out here too, there are two types of capillary cells, one of which of course was known and which we've heard about, and a second type, the capillary A cells, or aerocytes as I'll call them. Now, with these two cell types, capillary cell types, they were clearly distinguished, as you can see here, by clustering, and they separated from each other and for the other known endothelial cell types in the lung. They have a nice panel of markers that distinguishes the two types. And then when you take some of these markers and localize them, you find that both of them are localized to the alveolar region, and the cell types are intermingled in the alveolar region, as shown here, with about twice as many of the G-cap or general capillary cells as there are of these A-cap, or what I'll refer to as aerocytes. Now, fortunately, although many of the new cell types are not conserved in mice, these two cell types were conserved in mice. They are conserved in mice. And so you can identify A-cap and G-cap cell types and markers as shown here. And when we use these markers to localize these two cell types in the adult mouse lung, then what we find is, again, both of them localized to the alveolar region, and the two cell types are intermingled in the alveolar region. And so because these cell types are conserved in mice, we can use the power, begin to apply the power of mouse genetics to analyze their structures and their function. Let me just show you here, one of the most important things to do was to do a lineage trace. That is, we took the A-cap cells and permanently marked them with a lineage trace marker, TD tomato. And then we did the same thing in separate experiments with a G-cap lineage trace. And then we waited several months to ask, do these A-cap cells interconvert with G-cap cells or become other cell types? And the answer was no. These are stable cell types that remain the same cell type even after several months of a chase. So these are not rapidly interconverting cell states, but these are in fact true stable cell types. Now, with these kinds of approaches, we could now start labeling individual cells to begin to appreciate their structure. Here is the rather remarkable structure of a, sorry, I was supposed to be a movie here. Oh, yeah, of an A-cap cell. And these are remarkably expansive cell types, as you can see here, that they are hundreds of microns and usually span multiple alveoli. And remarkably, if you look closely here, these are cells that are filled with holes in them. They kind of look like Swiss cheese. And then each of the cheese parts of the cell are actually wrapped up on themselves and to form an autocellular junction, as a very tiny tube through which an individual red blood cell can pass. I'm looking at the next slide here. And what you see is you compare the A-cap cells to a single G-cap cell, and the G-cap cells are much smaller and much simpler and similar in structure and in gene expression to capillary cells in other parts of the body. And so we call them G-cap cell to say, because they are more similar to general capillary cells, whereas the A-cap cells, the aerocytes, are specific to the lung. Now, when we localized each of these cells using electron microscopy, what was remarkable was that the aerocytes shown here were immediately juxtaposed to the alveolar type 1 cells. They are the cell type, the capillary cell type, that forms the air-blood barrier. They form this intimate association with AT1 cells, the alveolar type 1 cells, the epithelial cells, and they form the air-blood barrier and mediate gas exchange. In contrast, the G-cap cells, as shown here, are never associated with one of these air-blood barriers. They are very close by, and they are associated with other stromal cell types, parasites, and fibroblasts. And so you can have these GCAP cells immediately next to an aerocyte, the GCAP associated with parasites and fibroblasts. And they form, we predict them to form a signaling and contractile unit with the parasites and the fibroblasts that regulates blood flow to the aerocytes. So they seem to be playing a key role here in controlling how much oxygen gets into the aerocyte, and then as blood gets into the aerocyte, and then is able to receive the oxygen from the alveolar lumen. Now, it turns out that these GCAP cells also, though, have one other completely unrelated but important function, and it's shown on this next slide here. These GCAP cells turn out to be the stem cells of the alveolar capillaries. That is, slowly over time during homeostatic normal life, GCAP cells will proliferate and give rise to aerocytes and also self-renew. And this looks amazingly like the diagram that I showed you for the alveolar type 2 cells, because it turns out that the GCAP cells as well appear to be facultative stem cells, fully differentiated cells that only moonlighted stem cells. That is, they regulate flow to the aerocytes by day and act as stem cells by night. And in particular, their stem cell activity is activated by injury to aerocytes and to other GCAP cells. And so there's this nice homeostatic regulation where you upregulate and call upon that stem cell activity under disease condition and times of need. Now, there's one other thing that's very much reminiscent of—
Mark, I can't even begin to tell you how absolutely gorgeous this is, but we're kind of out of time. And if you've completely sold me on the idea of a real hands-on workshop on this data. I noticed the incredible job that you guys did on the endothelial subtype annotation in your primary data. Could we have a follow-up workshop and actually get Kyle to be part of that too, and really get into some of these details?
Sure.
This is fantastic.
Can I finish now, or are we?
No, sure. Go ahead. Go ahead.
Oh, okay, great. Okay, so let me just thank you for that nice, Bruce. Greatly appreciate it. So the other thing that's very similar is that that both the GCAP and the aerocytes arise from a bipotent progenitor, and that at the end of embryonic and neonatal life, the GCAP cells take over the progenitor function. And you can show that the development from a bipotent progenitor, because you can label, at least in mice, individual progenitor cells in the alveolar plexus, and show, if you wait a month or so later, that individual cell has proliferated to give to multiple cells, as shown here, one of which is an aerocyte, and the others are all the GCAP cells. And you can, in mice at least, monitor the development, the morphological development, as shown here, from a relatively simple progenitor in the alveolar plexus. And you can see this remarkable maturation over the 2 weeks postnatal life. But the GCAP cells, you can see, do not grow or mature significantly in their structure, although they do molecularly, as I'll show on this next slide. We were able to actually reconstruct the full developmental gene expression program, going from the progenitor to the GCAP and the aerocyte cell types, by taking a look at the beautiful data set that Ido Amit's lab had generated a couple of years ago of the developing mouse lung, and to just re-annotate that and reassign fate based on this new knowledge, and basically work out then the full gene expression profile, including genes that are on only in the plexus, and then genes that turn on in each of the different cell types as they mature. And I think this is going to be one of the goals then of the developmental human cell atlas project. So let me just end here by saying that now we know that in addition to the alveolar side, on the endothelial side, there's one cell type, the capillary aerocyte, which is involved in gas exchange by directly opposing to the alveolar type 1 cell. And that cell type also looks to be a direct target for COVID-19, as I mentioned earlier. And then there's this other flow regulating GCAP cells that are also function as stem cells. And so here you see now the two sides of the barrier. And on the next slide, I'm just going to go get this back to COVID, because what happens is if the virus infects the alveolar type 2 cells and overwhelms the stem cell activity of the AT2S cell, they're then able to penetrate the epithelial barrier and start to reach the endothelial cells, where they can target it appears, the aerocytes here, and start to basically infect and destroy the aerocytes. And the GCAP cells now are a backup to try and keep up with the damage that's being done by the infection of the aerocytes. But if those stem cells are overwhelmed, if the virus gets the upper hand, then the serious consequences begin to occur. Vascular leakage from the blood into the lumen of the alveoli, basically the flooding of the alveoli, coagulation problems, and then the inflammation, that is the infiltration of the immune cells and the cytokine storm. And this is kind of what today's announcement has been, that perhaps inhibiting the cytokine storm might provide some benefit to these seriously ill patients. But we would love to know as well if there's things that can be done to recognize and to understand these earlier events. And I think this is a beautiful example of how complicated these disease processes can be. But with the precision of single-cell genomics and single-cell analysis, I believe we finally have the tools in hand to be able to understand, to begin to understand and ultimately treat these diseases in rational ways. And so,
Mark, bravo. Let's, we're gonna actually have to move on. I really, yes,
great.
Please, here's everybody. Thank you so much.
Yes.
Incredible. That was 3 talks, probably 3-hour talks in 20 minutes, maybe a little over. Anyway, let's go to the panel. We've got some great questions and thoughts, and I think actually you covered a lot of questions in the end of it. We need higher resolution. We need to see the EM version. We need to see those cell dynamics. All of that you started to bring out so beautifully. So thank you. Could we, maybe if we could step back for just a bit and then put this in perspective and then drill down a little bit. Aviv, do you want to try and talk about how development fits into the overall Human Cell Atlas project, and how?
I'll say a few words. First of all, thank you to the speakers. These were fantastic talks. And then I'll say 2 things. I think there's ways in which a developmental atlas and any atlas share features. We want to identify cells and their dynamic changes and their states and their interactions and spatial organization. But I think the thing that is unique about the developmental atlas is the way the change looms very large over a developmental atlas in every feature that you look at. So even things that are relatively well defined for us now, for example, the identification of discrete cell types, if we think about a developmental atlas, we have to also think about how the discrete cell types of one time point relate to events that happen later, as well as this ongoing change that will proceed even into adulthood with aging and so on, when sometimes you see some of the developmental processes. I think Dana is going to highlight that in the context of disease. So that's one component. And the second piece that I wanted to say, there cannot be a human cell atlas without a developmental cell atlas component within it. The two are inextricably coupled. And this is true because you cannot really say how these things emerged out of thin air if you only look at the adult. You have to relate to the developmental atlas. There are many developmental processes that continue, as Mark showed in his talk, that continue throughout life. And there is a mirroring of them in the earlier developmental phases. And then the third reason for that is that if we think of the atlas as a reference map, for example, in the context of disease genes, if you think of diseases like rare diseases, many of them have to do with manifestations that happen only throughout development. And then what we are left with is the consequences of those. And so that's why the developmental atlas benefits, I think, from many things that we would do in general, but also has to solve some problems that are much more acute in the developmental context. The morphological changes, the temporal changes, everything changes all the time in development. And so it highlights that. It's also true in adult, but in adult it's more subdued than it is during development. I'll finish with this and say what a fantastic start to the series.
Helen, Gloria, Sarah, do you want to talk a little bit more about likelihood of getting into rare diseases and informing rare disease underlying
I think it would be great if we could fit in. So I think it'd be great if we could fit in a short discussion around the genetics, and the rare disease specifically. And one question that could catalyze the discussion is basically to what extent the healthy reference cell atlas, that human cell atlas is primarily focused on, to what extent could that inform on rare disease? And it would be interesting to get a perspective from Helen on this, because clearly, the obvious thing that we all want to do is to look for the cells and tissues where the mutated genes are expressed, to look for where the mutations may be sort of penetrating. But that's I think that's a guide, but it may not always be informative on the disease phenotypes. So it'd be interesting to hear kind of perspectives from both Helen, Gloria, and anyone else who wants to chime in on that question, kind of in the next couple of minutes, and then we can maybe move on to pediatric tumors, for instance, and their relevance to development from a perspective from Donna and Muz.
I'll let Helen go first, since I haven't.
Yeah, I'm a clinical geneticist in Cambridge in the UK. I've really enjoyed the talks this afternoon. I think this is perhaps a two-way thing, in that a lot of rare diseases have quite subtle features that parents will often notice, but clinicians perhaps haven't appreciated so much. And I think there could well be hints that come from the cellular work about genes which are looking as though they're active, that perhaps linked to a rare disease, that will then reveal on deeper questioning of families that actually that is an attribute and a feature of that condition. And actually those children do have recurrent problems with respiratory complaints, which had just been parceled up as a sort of general feature of an unwell child, and people hadn't recognized were actually a very core feature of the disease. And I think that the Human Cell Atlas will really catalyze that sort of opportunity to really delve deeper into describing the full features of a lot of these rare and ultra rare diseases.
Fantastic, thank you, Helen. Would Gloria like to comment, and also any of the other panelists?
Yeah, I would largely agree with what Helen just said, right? So as we can go back and forth as we have symptomatology that we don't understand the etiology for, and we have identified additional cells or networks or mechanisms, we start to see where those may line up, right? You start to be able to define diagnoses, start to look at particular genes because we know something about their biology, and that they might be involved in the symptomatology that we see. And then, as Helen said, to be able to pick up more subtle symptoms, you get some clues, perhaps, if you know the gene or the cell that is particularly, involved in that genetic disease. The other is probably the polygenomic, much bigger task to try to tackle. But that is, many of our diseases are not single gene, but they're going to be multiple gene, or they're going to be susceptibility genes. And that will help us to get at not just the rare diseases, but some of our more common diseases, probably. So.
Awesome, thank you, Gloria. That makes a lot of sense. Yeah, I think the genetics will be interesting both for the rare and the common, to understand the contribution of development, both prenatal and postnatal. Could we move on, Bruce? Do you think we could move on to getting input from Donna Pear and Muzlifah Haniffa, because they've prepared a couple of slides.
Muz, why don't you talk about human development, and then also what its implications are for teaching us about how are we going to sample better? How are we, what level of sampling do we really need in order to get mechanistic in human development, understand human variation independent of the disease genetics, and then see disease genetics in the context of normal human variation. Just the robustness of these systems is spectacular, and that you can really have a disease effect in the context of the network robustness. How does that happen? What do we need to learn?
From the perspective of human development, is very interesting because of these temporal changes, and you have the same issue also with disease, because often with human, what you have are snapshots, which you then have to kind of integrate to get the kind of temporal information. And you have this challenge actually whenever you approach any disease, because you're approaching the disease, you don't know at what point of the disease progression that the sampling is happening. To make matters worse, during development, the time is so kind of like skewed, or I would say like half a day during early development is very different to half a day in postnatal life or half a day in adult life. So many things happen. So this idea of like, intervals of days doesn't make any sense, or intervals of weeks doesn't make any sense. You almost have to kind of have hypersampling very early on, and then a little bit more sparse, or you can afford to be more sparse later on. So I think this is all going to be extremely challenging. Add to the fact that there are a lot of three-dimensional morphological changes that also take place during development with all of the different cell types. So we've kind of come a long way really to unearth some of the complete unknowns, the black box of human development. And I think single-cell genomics has proven to be amazing in that sense, partly because we now have like a much better ability to identify cell states and so on and so forth. It was very limited in terms of the repertoire of what we could define for human development. And also the kind of like assumption that a specific marker or a phenotype that you see in adult, that same expression or whatever defines the same entity during development. So I think that's really this, the amazing power, and gradually trying to kind of like assemble all of these things. One of the things, the other point that I wanted to raise was with regards to the function, and it's very striking with Mark's talk about the kind of duet between the kind of capillary cells and the alveolar cells, the AT1 and AT2, and the equivalent of the A cap and G cap, and what those might have, the functions they may have during development, and the functions they may have during physiology, and the functions they may have in disease, and how that changes across the spectrum. I think I'll pass to Dana, who has some slides about human development, and she might be able to expand this a bit more.
Yeah, so I'm going to share a slide because I was asked to show pretty pictures, and a picture is worth 1,000 words. So I'm really interested in both development, the earliest forms, and cancer. They're sort of two sides to the same coin. And this is sort of one figure from adapted from a paper we recently published, where we looked at lung cancer. And primary lung cancer really activates a lot of the regenerative pathways that Mark talked about in later development. But what was quite striking to us is when we looked at the metastatic cells at single cell level, what we found is actually metastasis really recapitulates lung organogenesis. So we could find the gene programs for all the lung organogenesis, all the way from early endoderm and foregut, all the way to sort of the development of more distinct lung structures. And not only did we share the genes, they shared like really the same regulators, and one can really view metastatic cells as organs that are trying to grow wrong, which really shows how important it is to study development, because cancer actually doesn't really reinvent the wheel and take mutations to give it new abilities. It actually, many of these mutations give them access to really early developmental programs, and they abuse them. And, in the case of metastasis, try and build maybe a little mini lung in the wrong place at the wrong time in the wrong way. Another, pathway that one can look at that's very central to development and has been connected to cancer very early on is EMT. And we see that EMT pathways are often also on in lung. And here, not only do we share the same transcription factors in lung cancer, but also the same signaling pathways. And EMT in cancer is considered something bad, but in development, it's very organized, it's very structured, it's very controlled. It's used again and again to do the morphogenesis, to do the movement, to create the shape of our body. And actually in cancer, often we find a regulator of EMT or a regulator in cancer, and then we go back to development and discover that it's important to keep the controlled, orderly process. And again, EMT is important to rise again, because development is so dynamic and you have so much movement and morphogenesis, and that's going to be one of the bigger challenges for us to do this atlas. And finally, since I was asked to talk also a little bit about pediatric cancers, I didn't really prepare that because lung is not a very pediatric cancer, but one of the key fundamental features of early development is that cells are very plastic. They haven't sort of matured, they can make many decisions. And one of the features of cancer is actually increased plasticity. And in pediatric cancer, many of the regulators that are mutated are actually epigenetic regulators. And what cancer does is it sort of endows cells with plasticity that's typically connected to development. So, if we want to understand cancer, we have to get to development. We have to get to very early development at the very beginning, gastrulation, because that's what sets the stage. And we have to understand not only the cell types, but how they move and organize and migrate over time.
I just wanted to add something to what Dana has said, and that's this hijacking of developmental programs in cancer and so on and so forth. And I think this probably is a bit more generic than just cancer, and you can probably see it maybe in other kind of like inflammatory disease settings and so on and so forth. And it might not just be the kind of like epithelial programs, but also the kind of immune programs that may be hijacked during kind of like cancer-related immunoregulation or immunosuppression. And this is completely uncharted, and will be very interesting whether these programs are actually developmental programs.
We see that all the time. For example, again, breast cancer cells like to hijack programs from macrophages to metastasis and say, "Hey, I'm a friend. Don't get mad at me." And they actually express macrophage genes, which are supposed to signal, good things. Yes, there's again, I study cancer, so I see it there. But there is pervasive programs of hijacking of programs of all cell types, not just epithelial cells. It's very central. So if we understand these programs, particularly the developmental and regenerative ones, we'll have a really great handle on cancer. And also looking at mutations that recur in cancer, we can map back to what might be important in development.
Gary Bader, I'm wondering if you would like to, we don't only have a couple more minutes left. I wonder if you'd like to chime in here, talk a little bit about integration of many data sets. I think that's a part, sort of an
I've answered that
question.
online. I've answered that online. So
okay.
I think we can move on, yeah.
Talk about integrative pathway biology, integrative human cell atlasing, how many people do we need to survey, and then put it all together that we can provide the community with research tools, data tools, and that's going to transform to knowledge that's going to help people.
Maybe if I could just mention one thing before that is maybe related to the last point about helping people, is also the point that Dana mentioned about regeneration. So the regenerative medicine area is also an important one that that can really learn from developmental atlases if we're going to grow cells and use them for tissue repair. I don't know how, it might be interesting to have more discussion in later time about how those can be applied in the context of the clinical problems that Gloria mentioned to actually fix the rare diseases once they happen. Obviously there's gene therapy, but there might be different ways of doing it. But clearly, all of this is a huge the developmental atlas is a really massive project. We're talking about the adult atlas, but the developmental atlas is sort of many adult atlases because it sort of changes over time, and there's so many radical changes that happen over time. So each of those time points need to be mapped, and they all need to be put together. So we really need to energize a lot of different people to think about how that works, from developmental biologists and embryologists and physicists and computational biologists and many others to understand these processes. And then that will have obvious applications for these diseases and repair therapies that we've been talking about in this panel. I don't know if that answered the question, Bruce.
Those are great answers and great points. I mean, and I think, just to plug for the Human Cell Atlas and HubMap and LungMap and efforts like that, that just the way that the data coordination centers are trying to work together and trying to put more information together. So these data sets are connected and cross-informing. It's fantastic, and we need more people. We need the community to use that to help with it and to lead new efforts into doing that, because nobody is going to be able to look into all the important areas that are necessary to inform human health.
So should we show the last slides if it's 1 o'clock?
Sure.
I think that's a fantastic point to wrap up, Bruce. That's a great point, and I think we just have a few slides to wrap up. If Christine
maybe
could
point
share
out
that.
to people before the wrap-up slides. Maybe point out to people that Mark and Gloria agreed to stay for additional time after 1 p.m. for those who want to engage in Q&A with them live, for which we're extremely grateful. I want to say that before people start signing off. In case they wanted to hang around too.
Christine, were you going to share the slides? I can, if not. Okay, so we're at 1 o'clock, and that was the scheduled time that we agreed to end this seminar. So just in the to respect everybody's schedules, we want to end this now. And as Aviv mentioned, the speakers will stay on to answer questions. So we just wanted to thank all the speakers, and this program is being. We're going to have a number of different seminars as part of this program. There's a number of people behind us, Bruce, myself, Alain, Maz, Stan, Aviv, Deanne, and Sarah have really thought about how to keep things going during the pandemic, keep scientific discussion going. Christine, Tracy, John, and others as part of the HCA Executive Office, and Scott, who did the AV for this, we want to really thank. And we wanted to mention that so thanks also to the speakers, of course, and the panelists, especially Helen for joining. Next time it would be great to get feedback about how this process worked from everyone and how much time we have devoted to the different segments of this. The last couple of things are just to save the date for Human Cell Atlas Development and Pediatric Atlas meeting that we have rescheduled from this July to July 2021. We also wanted to mention that in the meantime, we can also discuss Human Cell Atlas Developmental Atlas mapping projects at the Company of Biologists Stem Cell to Human Development meeting in September. It's a virtual meeting. And finally, we wanted to just mention that the next developmental seminar will be on spatial transcriptomics with guest speakers Ali Ertuk and Mats Nilsson around mid-July. Stay tuned for the exact date. And if you want to just keep in touch with this discussion, these links are made available, and we'll be sharing them as well after the meeting, along with a request for some feedback about how we did on this inaugural webinar for the Human Cell Atlas, Developmental Atlas, and Pediatric Atlas maps. So thanks very much. I guess we for everyone who wants to continue to ask questions to the speakers, I know there were many questions that were asked in the Q&A session, and we also, the speakers, Mark and Gloria, have been answering many of those. But Mark and Gloria, do you want to just take over the floor and answer questions live or by typing as you wish?
Sure, happy to do whatever.
Yeah, I'm open for questions. Yeah.
Mark,
I think
we can start with, yeah.
Mark might have had a couple that he was planning to answer live.
Okay. Yeah.
Let's see. Actually, there was one that I hit that button by mistake.
Okay.
I sorry about that. I thought that maybe was for you. Was about the role of inflammation in lung aging and the phenotypic. I thought that might have been for you. I wasn't sure. From Shankar. Is that what you're talking about, Gloria?
Yeah, I think it might have been. Now, can I find the question?
I mean, it's certainly related to the effect of COVID on lung state, and what's mild versus severe and inflammation look like. How does the lung handle these robustly inflammatory challenges and regenerate, or how does it add up to a lung that undergoes interstitial fibrosis? Or
yeah, so there are clearly developmental aspects of the immune system too that we are trying to get a handle on, right? What immune cells traffic to the lung at what ages, whether it be prenatal or postnatal childhood and to adult, right? And how they interact with the parenchymal cells of the lung. Other investigators are really starting to put out that kind of information. So, we do have indication that the early inflammation in the lung, early response to oxygen, for example, is going to have longer term effects on the alveolarization and on fibrosis. And probably, I mean, we know that early lung infections, like with RSV, have a very different severity of illness and influence the response to those infectious agents later. So I'm not sure if I'm really answering your question very well, except that fetal and childhood neonatal inflammatory responses in the lung can have long-term effects, and we're starting to get at the individual cells, characteristics of those individual cells that might be responsible.
Thanks, Gloria. I should have mentioned for the panelists, if anyone has to go because we are over time for the scheduled time, I know some people might have had meetings to go to, feel free to go, and anyone else who wants to stay, we can continue this and answer the questions from the audience. Mark, there were a couple of additional questions for you as well.
Okay, let's see. I'm looking at one now from Ravi Misra. I have a question about the spread of the SARS-CoV-2 into the alveoli. Has anyone in the greater scientific community addressed whether or not macrophages are actively bringing virus to the alveoli? This has been suggested to be a mechanism for severe influenza infection. That is a really interesting idea. The macrophages have direct access to the incoming virus, and then once the the epithelium is breached by the virus, there's plenty of opportunities, not just for the alveolar macrophages, but for other monocytic macrophage and dendritic lineages to engulf. I think bringing it to the alveoli, I don't know of any good data on that, and I'd love to know just some direct effects of virion particle, the interaction between virus particles with alveolar macrophages. We're trying to, isolate each of the different lung cell types and directly expose them to purified virions to get at least an initial answer ex vivo on that. So it's a great, excellent question.
And Mark, looks like there's another question, I think.
Okay. let's see, I see one here from Suvarna Gandur. For the deeper profiling on your cells using SmartSeq2, what is the sequencing depth you use versus that for 10x? Yeah, we're usually going up to something, it varies on experiment to experiment, but we're usually going up to like 500,000 reads. We get saturation, we start to get saturation around there on the SmartSeq2. And so that's kind of the depth we've been trying to achieve typically in those experiments. 10x, we're getting saturation 10x more like at 50,000, something like that, 50,000 reads.
And what are you thinking about Nuc-Seq in the lung? And are there some cells that it doesn't work well for using Nuc-Seq, and sort of getting into old frozen sections and that sort of thing to go back and reconcile the histology versus the samples?
Yeah, so for the Nuc-Seq, we have not experimented a lot so far with Nuc-Seq. One of the reasons is because we've been wanting to use surface markers, available surface markers, to balance the population so that we can get a nice distribution of cell type across all of the major tissue compartments. For the fragile cell types, that is, particularly valuable. So that's a nice idea. And then thinking about using it, in preserved specimens and how to access preserved specimens. So far, we haven't experimented too much with that, except for we've been starting to do some of that with neurons because of their fragility. And there are a few other lung cell types that are fragile in our hands. And actually, interestingly, SmartSeq2 and I don't know if other people have found this, but SmartSeq2 and 10x, there are idiosyncrasies between those platforms in terms of certain cell types being able to harvest and analyze them, capture them for sequencing. And so it's interesting that there are platform-specific idiosyncrasies. So one of the unexpected advantages of using both platforms is we captured some cell types in one platform, but not the other. And that clearly wasn't, exclusively due to abundance of the cell type. It has something to do with fragility. So presumably, Nuc-Seq is going to be less sensitive to those types of issues. But one of the thing that we've been going after are cell types that are extremely rare and haven't been captured at appreciable levels or not at all so far. I mentioned several of them in my talk in our bioRxiv paper. And so for those, I think we've been trying to develop enrichment strategies, trying to figure out surface molecules from known expression profiles or good guesses to see if we can enrich for some of those. So I think we haven't yet turned to Nuc-Seq, but it'd be nice to hear what other people think of those advantages and disadvantages, to compare and contrast.
It's an interesting question with technologies versus cell type capability to see the individual cell types. And there's been a couple of recent studies, Aniko and Fabio and Ravi, where you have you're seeing inflammatory cell dynamics, and on the other hand, the ability to see those cells in context with 10x technology isn't necessarily where it needs to be. I don't think. And I'm wondering, is that possibly non-polyadenylated? Are we going to need new technologies to see that those kind of cells using transcriptomic profiling?
Mark, can I ask about the duet that you proposed?
Okay.
At what point during development or kind of like postnatal life that these stem cells become facultative stem cells? And what do you propose they were doing during early development when the kind of lung environment was actually more aquatic rather than ventilated?
Yeah, I think that whole transition, molecular and functional transition, is something that needs to be studied, in great depth. I think having the expression profile for how the individual cells are changing. Although one of the things that makes it a little bit challenging is there's a lot of local heterogeneity in any developing lung sample. That is, at certain places, positions in the lung, like alveolar development is more advanced than at other places. And so what that means is that you have to, knowing what stage they are at, you either need, very precise morphological markers or, better yet, molecular markers. And so the limited markers so far, that have been used, or, I think basically we need some spatial transcriptomics to kind of get at that. I mean, the advantage of that temporal heterogeneity and spatial heterogeneity in the lung, it meant that, and I think it's significant for the Atlas projects, for the developmental Atlas projects, is you can take advantage of that heterogeneity, and that from one sample, by doing single cell RNA sequencing, you get multiple different stages of the developmental process. And so, you can use that, reconstruct developmental pseudotime from that kind of heterogeneity. And so you don't need to get, precisely every hour of the developmental process in order to get good gene expression profiles, because you can take advantage of that heterogeneity. So what they're doing earlier, and for like the alveolar type 2 stem cells, that they are not active for about a month or a month and a half after birth. You see very little activity. And so there seems to be a period at that time when the bipotent progenitors are being depleted or very difficult to detect. And then the handoff, there might be some kind of a gap, or might be some kind of intermediate stem cell that hasn't been identified that plays a role in between those phases. So I think that those trends, it's an interesting question, and to see what the environment, the very different environments of the lung in utero versus ex utero after birth, how those contribute will be interesting and fruitful avenues to pursue.
So we have quite a few questions still coming in, and there's still about 170 people participating, which is really great. I'm just wondering how much time the speakers have available, who are already generously providing their time for this.
I think 10 or 15 minutes is fine for me.
Yeah, I'm okay. Yeah, I'm okay for a little bit.
Okay, so half past the hour, we'll say.
Sounds good.
Sounds good.
Yeah, I don't know, Bruce, if it was one of the questions that you were asking, but how do we get at not only rare cells, but rare transcripts, right? Because many of those immune transcripts, or the transcriptional factors that are going to be important program regulators, are of such low expression at any one time that we exclude them, right? We don't.
I'm not sure that they're so low so much as they might sometimes be coming in if they're non-polyadenylated, and a lot of AP1 downstream, ATF downstream transcription comes in non-polyadenylated for very short-acting proteins. Sometimes those are the most important effectors. You'll see them on protein-based assays, but I think poly-A-dependent technologies aren't seeing them. Particularly this, immune adaptation, inflammatory adaptation, very short, rapidly important things. I'm not sure where we are on that.
Do we have technology to address that?
The CyTOF versus single cell, maybe. Mark, do you have any ideas on that for, particularly in the COVID stuff? Are we really seeing the complexity of those immunoeffector cell types?
Well, I should say one of the things that we've looked at very carefully is the sensitivity of single molecule FISH versus, SMART-Seq2, our best way of getting, detecting rare transcripts. And I should say that when single molecule FISH is slightly, in our hands, is slightly more sensitive, like RNAscope and related technology, is slightly more sensitive in our hand than SMART-Seq2, so that it looks like when you get down to 1 to 2 transcripts that are detected by single molecule FISH per cell, that is getting down to the level where you're starting to get a lot of dropout in the SMART-Seq2. And so, I think that the single, and then the question is, what's the rate of, dropout, for single molecule FISH, and so how low, can they go? How sensitive can each of these techniques go? Because I do agree that for interesting transcripts, we would like to be able to go down to those detection levels. Some of the ones that we're seeing 1 to 2 transcripts, for example, are, hormones and neuropeptide genes, and obviously would like to know the full complement of those that are being expressed, as well as many other genes.
As single cell ataxia starts to cross-inform single cell transcriptomics, or can we see those poised loci through that approach, and then see the potential there?
Yeah, that seems. I think that would. I think that makes sense. Kind of going back to the chromatin. If that's, I haven't looked. Do you think that is much more sensitive than single molecule FISH? I mean, in terms of getting at active loci, obviously you can get to pre-active loci, but in terms of getting to active loci, do you have any feeling for that?
I mean, my feeling is it's going to be there. I think Shinsun's got some cool results coming in that area. That those two switches that you had in terms of the pre-type 2 cell that was going to go to type 1 or type 2 that's poised for regeneration. Same with the endothelial cell precursor. I think if we could get at their epigenomes, they should show sort of the union of both alternative programs, and then it's a question of what's the signal and transduction that gets you then daughter control.
Right. I think that is fairly straightforward for the AT2S. If those are the stem cells, which is our best guess at this point, although one of the really interesting things about the AT2S cells in human versus the AT2 stem or AT2 star in mice is that the AT2 S in humans, they show many differences. They show many differences in gene expression from canonical AT2 cells, whereas in mice, the AT2 stem, beyond the axon 2, there are very few gene differences, between them and canonical AT2 cells. And so they don't cluster out. In our hands, they don't easily cluster out. You have to go to great lengths and purify the axon 2 positive ones and look at their expression profiles, because you can't just do random cells and begin to distinguish them. But for the human AT2S cells, they are transcriptomically different, and so they are easy to identify. And so presumably, understanding some of the chromatin changes and the other factors that are preparing and how they're contributing to their stem cell activity will be very interesting and important if in fact they are acting as the alveolar stem cells, which is right now just a hypothesis.
And how are they different from the developmental equivalents?
Yeah, actually, so that's interesting too. That the developmental equivalents, are very different, and that's true for the AT2S, and that is true for the capillary, the GCAP cells compared to the progenitors, even though their structure looks kind of similar and things that, as I showed with the developmental expression pattern analyzed from reanalyzing EDO Meat Labs data from mouse, it's not like the bipotent progenitors are the same as the GCAP. They each have their own expression program and markers, and so I consider them two different cell types, if you will, certainly two different molecular types.
Great.
Actually, I got a question here from Ron Germain. He's been asking because, hi Ron, if you're still on, he's been asking about how many markers does it take to distinguish all of the cell types, in a human lung, the 58 molecular types in a human lung, so that, for spatial profiling, for example, what's the minimum number of probes that are necessary? And in the bioRxiv paper, we were identifying the optimal markers, as I briefly showed in my presentation, and then asking, what's the minimum number to get, virtually all of the molecular types in the human lung unambiguously. And it turns out to be something like around 150 probes, because it's very hard to find just a single probe across the entire, that's specific for a cell type, across the entire 58 molecular types. And so usually you need 2, and in some cases 3 probes in order to molecularly identify the molecular type of interest unambiguously. So that's for people who are interested in doing spatial transcriptomics and you don't have yet access to 100, 1,000 plexing or something. You can get by with well-selected probes, about 150 or something like that, to do a quite good job for spatial transcriptomics. Let's see. Somebody's asking about spatial transcriptomics. I don't know if anybody wants to answer that one. Is there any spatial transcriptomics methods being used by the HCA that allows a resolution of one cell, like 10 microns or less? I think that is a great question, because somebody else can weigh in on this. I feel like it is so important to get down to single cell resolution, because when you're doing single cell RNA sequencing, for example, and you get a little contamination of a neuron, for example, with a tightly associated glial cell, so you don't have complete cellular resolution. The utility, that has some utility, of course, but not nearly as much as having the purified cell type. So I feel like that is spatial transcriptomics, will be mature, but it has to, it's coming mature, but it really has to get that single cell resolution in my mind.
And the next session in this series will be about spatial transcriptomics, so that will be a good place to discuss that in more detail.
Yeah. I think that's a wonderful transition to that session. Sina Busagi is asking, with which technology did you find more cell types? We found more cell types with the SmartSeq. We found more cell types with the 10x, but mostly that was because we profiled so many more cells with that. So that has kind of an unfair advantage. But in terms of resolving cell types, there were quite a number of cell types where the SmartSeq2 and the deeper expression profile coverage, even with the more limited numbers of cells, was able to distinguish molecular types, whereas it was not so obvious, with the 10x data. So that quality of data. So basically, the way that we do it to balance, cost, and effort, because the SmartSeq2 is about 10 times more expensive in our hands per cell analyzed, and much more work, is that we end up doing about 90% of cells and 90% of our resources towards 10x profiling, and 10%, 10% of cells to SmartSeq2. Actually, that ends up being a kind of a balance, an even balance, because of the different cost per cell. We do 90% of cells by 10x, 10% by SmartSeq2, and that ends up being kind of a balance in terms of allocating resources, about half and half between those approaches.
How good is the SmartSeq2 at resolving alternate splice forms? And are you thinking of even longer read sequencing as giving you a scaffold for better interpretation of the isoform patterns? Yeah,
the SmartSeq2 is very good for the splice form. So we've been doing a deep dive into alternative splicing, and you get some information, some useful information, from the 10x data. Of course, near the 3' end, the poly A tail, if you're doing 3' priming is always, is always best, but also because you get reads that are internal, fortuitously get reads that are internal. You also get some coverage there. But the SMART-C2 has been very good for that, and I think that is an important frontier that has not been exploited, to the way it can be. And I think part of that is because a lot of the droplet-based methods are not as good, and so it's a little bit more expensive and painful to collect the SMART-C2, where the alternative splicing and alternative splicing is better. For us, we've been doing this non-human primate model, the mouse lemur we've been developing and doing organism-wide atlases. There, it's been particularly important because, the annotation of the genome, is not, of course, as advanced as it is in mouse or human, for example. So there's a high quality, of course, genome assembly, but identifying and annotating genes, by phylogenetic approaches and some limited amount of bulk RNA sequencing. It's been nice. We've been really taking advantage of using 10x and SMART-C2 to not only identify expression levels, but to map the structure of genes on the genome. So that we've been kind of pushing that, because I think for new model organisms, the non-canonical model organisms like mouse lemur, it's really a valuable single-cell RNA sequencing is really a valuable approach, not just for identifying cell types, but identifying and molecularly characterizing genes and alternative splicing.
So it's 1/2 as we mentioned. Should we end here? I know there are quite a few more questions, but we'll probably be here for another hour if we discuss all of them. Does anyone want to say anything in closing? Any last words?
I'd like to thank Christine Schwenk for the amazing job she did of getting us organized in such a quick amount of time for this conference. Christine, thank you. You're amazing.
That's very nice of you. Thank you.
And
Okay.
Mark and Gloria, thank you guys so much. Fantastic. Yeah, definitely we could go the whole afternoon, and we should again.
Thanks, Bruce. Thanks to you and your
Bruce,
organizers.
Yeah, I appreciate your organization, Bruce, as well.
Christine
Really.
and Gary.
Well, but bringing the ideas together, right? Recognizing the ideas need to be talked about. Yeah. Thank you.
Everyone.
Muzz is actually our secret ingredient.
And I just wanted to thank everyone who's attended the virtual conference today, and the speakers, Christine, and the panelists, Bruce chairing. It's been fantastic. Thoroughly enjoyed it, and it will be the start of many and more exciting virtual seminars to come.
Excellent.
Okay, great.
Thank you.
Thank you, everybody.